Hi, this is Elodie for the Massive team, and I'm going to show you quickly how you can use our web server ASES to visualize the protein diversity generated by alternative splicing in evolution. So this is the web server. Basically, you have a space here to query a specific gene. So you can put the name of the gene or the ensemble ID. Here you have the species where the gene is from. And then you have different parameters that you can define. So basically, the web server unites two methods that we have developed for assessing alternative splicing conservation. These are thorax and philosoph, and you can change the parameters for these tools here. You have to define a list of species that is of interest to you, where you're going to query the gene and see the different transcripts that are in these species. So here I have a set of 12 species that go from human to nematodes. And what I'm going to do is to launch, just to load the example that we have, which is the MapCan main example. So you can see that this is the result page. It looks like this. So you have an idea that the retrieval of information from ensemble, thorax, and philosoph succeeded. What you can see here is on the left, the evolutionary splicing graph computed by thorax. On the right, the phylogenetic forest of transcripts computed by philosoph. So basically, each node here is a transcript. So for example, I have one transcript in the most recent common ancestor of all my species. If I click on it, I can see its composition in S exons, which are exons or smaller parts of exons that are conserved in evolution. And so the transcript represents a path in the evolutionary splicing graph. So here, each node is S exon, and I have the transcript, which is a path. Okay, so I can see that this ancient transcript, the red transcript, corresponds to this path in the graph from here to here. So I can show the start and stop node, for example, for convenience, or not show it. So then what I can see here is that I have also one transcript in zebrafish and one in platypus. And in the ancestor common to Eutheria, there are two more transcripts that arise, that appear and remain fixed thereafter. So I have the blue transcripts, which correspond to the skipping of this part of these S exons here, and the green transcript, which has the exon 10-0, instead of 10-1 in the red transcript, right? So I can see I have a pair of mutually exclusive exons here, and also I can skip those exons. And basically, this is the most conserved alternative splicing event I have in the evolutionary splicing graph. So these three transcripts have evolutionary ancient origin. So now I can look a bit more at the amino acid sequences of these S exons in these transcripts. So if I click, for example, of 10-1, I can see at the bottom the sequences that I have in the different species. And what I can see is that it's very conserved, right? So I have all my species here, and well, basically it's almost 100% conserved. I can also compare this S exon with the other one, 10-0, which can be exchanged. And I can see that both look very much alike. So they're very conserved and very similar. So now I can also look at a specific transcript. For example, I'm going to take the third transcript of cow. And again, I have the amino acid sequence. So basically, I have access to the proteoform of corresponding to this transcript. And I can specifically see the path, again, in the evolutionary splicing graph, and I can query specific subsequence. So for example, I want to see where is this S exon in the sequence of the proteoform. So I'm going to just query DIWSV, and I can see, okay, that it's more or less in the middle of the proteoform. Okay, so then if I go a bit down, further down, I can see now a genomic map of the genes that I'm considering. And again, the boxes represent the S exons. So for example, if I take the same example of S exon 10-0 and I want to see where it is, I just have to click on this box here. And this is the orange S exon, the orange boxes that I see. So I can see where it lies. I can also see where is the other one that corresponds to the mutually exclusive pair. So this is very convenient to have also the gene structure. And Then if I go further down, I can have access to all the information regarding all the ASES and the transcripts and of the alternative splicing events. So for example, I can see that the first event, which is the most conserved, top ranks correspond exactly to this exchange between 10.1 and 10.0. It's an alternative type of event. I can see the conservation that I have in the different species and the species that contain this event, canonical path or alternative path, and also the consensus sequence of the canonical and the alternative ASES. Okay, so I hope that I convinced you that ASES is easy to use, that you can have a lot of information, that you can really move between the different representation and make some connection between these representation. And I hope you will be happy to use it. Thanks.